Okay, so today we are going to study the topic link reaction. Now, giving it an overview of the topic. In the previous topic, we studied that aerobic respiration consists of 4 stages, which include the first one is the glycolysis, which we studied in detailed in the last class. Then we have link reaction. The third one is Krebs cycle. And the last one at which the aerobic respiration gets completed is oxidative phosphorylation. So, as we can see here, that link reaction is in the between of 2 processes, 2 stages of aerobic respiration, basically glycolysis and Krebs cycle. Now, as the name of the link reaction suggests, it links the 2 processes, that is glycolysis and Krebs cycle. How does it link? It uses the product formed in the glycolysis and then forms a product which becomes the reactant of the Krebs cycle. Now, let me show you here. What's happening here is, as we can recall, that the product of glycolysis was pyruvate, which is a 3-carbon compound. Link reaction uses this compound, 3-carbon compound pyruvate. Well, what is the location of this link reaction? We can clearly see in this illustration, it shows that glucose enters into glycolysis. Glycolysis forms a product called pyruvic acid, or we can also name it as pyruvate. So, pyruvic acid, the place where this glycolysis happened was the cytoplasm of the cell, right? Now, pyruvic acid from the cytoplasm, it enters into the mitochondria, the mitochondrial matrix, where it takes part in the link reaction. So, the pyruvate enters into the link reaction. Now, what's happening in the link reaction is that this pyruvate molecule, it undergoes 2 things. The first one, as we can see here, is production of a carbon dioxide. Pyruvate is a 3-carbon compound. It has to remove 1 carbon atom. Now, this 1 carbon atom gets removed in the form of carbon dioxide and produces a molecule of carbon dioxide. This removal of carbon dioxide from pyruvate is known as decarboxylation. So, first thing what happens to this pyruvate molecule is decarboxylation is taking place and a carbon dioxide molecule is formed, which removes 1 carbon atom from the pyruvate molecule. So, now the second thing what happens here is that this pyruvate molecule removes a hydrogen molecule. This hydrogen molecule gets attached, it gets combined with the hydrogen carrier molecule NAD, nicotinamide adenine dinucleotide, and it gets reduced, forming its reduced form, reduced NAD. So 2 things happen here is that first one is decarboxylation, formation of a carbon dioxide molecule, and then dehydrogenation is taking place. Hydrogen molecule is getting removed from the pyruvate, which gets combined with the hydrogen carrier molecule, forming a reduced NAD. Now, as 1 carbon has been removed from forming carbon dioxide, this 3-carbon compound removes 1 carbon, forming a molecule called acetyl, which contains 2 carbon atoms. So, in the end of the link reaction, a molecule called acetyl group, which is a 2-carbon compound, it has been formed. Now, what happens is, till here we have used the product which was formed in glycolysis, that is pyruvate. We used this molecule and formed a new product from the link reaction, that is acetyl group. Now, how does this product in the link reaction becomes the reactant in Krebs cycle? So, what we have to do is, this link reaction forms a compound acetyl group, which is a 2-carbon compound. Now, what happens is a complex molecule, that is coenzyme A. It comes from the matrix, mitochondrial matrix, and it gets attached to the acetyl group, forming a compound named acetyl coenzyme A. This is the reactant of Krebs cycle. It enters into the Krebs cycle. So, what was the role of coenzyme A? Now, giving you some information about this coenzyme A, what is it and how it is formed. Here, coenzyme A is a complex molecule. It is composed of a nucleoside and vitamin. Now, roughly drawing its structure so that you can memorize it easily. A nucleoside consists of a ribose sugar, that is a 6-carbon containing molecule, and a base attached to it, that is adenine. Adenine base and ribose sugar. These 2 together is called nucleoside. Now, what happens is, to this nucleoside gets attached another compound, which is a vitamin named pantothenic acid. This vitamin gets attached. These three substances together, combined together, they form a molecule of coenzyme A. So coenzyme A consists of ribose sugar, adenine base, and a vitamin that is pantothenic acid. Now, what is the role of this coenzyme A? Why does it get attached to that acetyl group? So basically, this coenzyme A acts as a carrier molecule. In order for this acetyl group to enter Krebs cycle, this carrier molecule takes the acetyl group to the Krebs cycle. So it acts as a carrier molecule. It carries this acetyl group to the Krebs cycle, so that the next cycle can be proceeded. Now, summarizing linked reaction. Linked reaction, first of all, the pyruvate passes through the outer and the inner mitochondrial membrane via active transport from cytoplasm into the mitochondrial matrix. Cytoplasm, it has been formed in the process of glycolysis, which takes place in the cytoplasm, and then it enters matrix so that it can take part in link reaction. Within the matrix, what happens is, first, decarboxylation, that is removal of carbon dioxide, takes place. Then dehydrogenation, that is the removal of hydrogen, takes place. And third, coenzyme A gets attached to the acetyl group, forming acetyl coenzyme A. Now, what happens to the hydrogen that has been removed? The hydrogen removed from the pyruvate molecule, it gets added to the hydrogen carrier molecule nicotinamide adenine dinucleotide and forms its reduced form. This equation shows the whole process of the link reaction. The reactants are pyruvate, which is 3-carbon containing compound, a coenzyme A, and hydrogen carrier molecule NAD, forming acetyl coenzyme A, carbon dioxide molecule, and reduced NAD. Now, as we discussed earlier in the last class, that we can have substrate sources of energy to produce energy other than glucose. Now, what will be the sources other than glucose? It can be fatty acids. So, if we have a compound other than glucose, which is being used as a source of energy production, where does it take part? It does not undergo glycolysis. Instead, because in glycolysis, we only need glucose, which can be broken down to produce pyruvate. So, we can replace this pyruvate with other compounds. At this place, we can replace the other compounds like fatty acids can be used instead of pyruvate. Fatty acids directly take part in the link reaction and form acetyl coenzyme A. So when we have sources other than glucose, glycolysis does not take place, because in glycolysis we need glucose that can be broken down to form pyruvate. So fatty acids that have been formed from fat metabolism, they can also be used to produce the acetyl coenzyme A. What happens is that these fatty acids, they undergo a cycle of reactions in the mitochondria. In the mitochondria, the fatty acids undergo a cycle of reactions. Each fatty acid gets broken down into a 2-carbon containing acetyl group. This acetyl group simply combines with the coenzyme A and then forms a reactant, which can be used in Krebs cycle as reactant. So this is the whole about link reaction. Inshallah, in next class we will continue with Krebs cycle.